on to our next speaker, JonasTreebak from here. Very excited to have you here.
Thank you. Is the microphone on? Oh, good. All right. So thank you very much, Morten, and thank you to all the organizers for pulling off such a great event. It's a pleasure to be here today. I will be talking about the effects of nicotinamide ribosides and pterostilbene on regeneration of aged human skeletal muscle. So this picture here shows a recent experiment that we did with cardiotoxin in mice. And I like this picture very much because it's depicting sort of the intersection between what is previously damaged and what is undamaged skeletal muscle. And the previously damaged muscle here shows many of the hallmarks of regeneration. We have the smaller fibers, we have the centralized nuclei, the infiltrating immune cells, and also fibrosis. And it also kind of shows the process. So we are going from here and on up to the fully recovered muscle. And obviously, what is needed to regenerate muscle is our muscle stem cells. They are located under the basal lamina of the muscle fiber. And in response to damage or injury, they become activated, they start to proliferate, and they eventually fuse with the existing myofibers. And our interest in this process, now it works, is actually what is the role of NAD in this process? What can we boost part of these processes or the full process by providing NAD precursors, for instance? Just a quick background. I know you know some of and also from previous talks you've heard about NAD, but NAD is this cofactor involved in redox reactions in the cell, where it's continuously oxidized and reduced. But it's also a substrate for multiple reactions in the cell, regulating cell division, DNA repair, chromatin remodeling, and mitochondrial metabolism. So being continuously used as a substrate, being continuously degraded, we need to have biosynthetic pathways in the cell that regenerate NAD, and these are depicted here. There are a few more, but the main ones here is the NAD salvage pathway controlled by NAMED, starting with nicotinamide. There's also nicotinamide riboside, nicotinic acid riboside, and tryptophan, and a few other NAD precursors that are also relevant. Our studies have mostly been focusing on NAMED. We've generated tissue-specific and also satellite cell or muscle stem cell-specific NAMED knockout mice, and we're trying to rescue any defects that we see with precursors. I won't be talking about the preclinical work today, though, but just to highlight some of the things that we've been doing. So there is a sort of a general consensus in the field that NAD declines with age. And I think this is kind of confounded, to be honest. If we took a deep dive into the literature and wrote a review about it quite recently, and it's very few cell types and tissues across species that actually show a decline with where the science behind it shows a decline in NAD. One of the tissues where you actually do see a consistent decline with age is skeletal muscle. So as we age, levels of NAD decline in muscle, and we've also shown that if you exercise train older people, you can sort of rejuvenate NAMED levels. You can increase the capacity of the muscle to maintain NAD levels. So what about muscle regeneration, and can we stimulate that by providing ARDD precursors? There are a few studies out there. The previous speaker has published 2 of those. I want to highlight one here, where the Johanovax group showed that by providing ARDD precursors, they could increase ARDD levels in the muscle stem cells, both in young and old. They could increase the number of muscle stem cells, both in young and old, and the ability to regenerate from cardiotoxin-induced injury was also increased here, measured as both histologically and also looking at embryonic myosin heavy chain expression. Another compound that we have been interested in is pterostilbene, which is basically methylated resveratrol. So it's a dimethylation compared to resveratrol. And there are also a few studies on the effects of pterostilbene and tissue regeneration. 2 of these studies actually show that you can enhance autophagy with pterostilbene, both in liver and muscle, and that this may play a role in regenerating the tissue. We're also interested in this because the company that provides the nicotinamide riboside for our studies, Elysium, are using a combination NR together with pterostilbene. So in the clinical study that I'll show you data from, we've used BASIS, which is a combination between nicotinamide riboside and pterostilbene. So we hypothesized that the dietary supplementation of NRPT would affect muscle regeneration after muscle or contraction-induced muscle injury in elderly humans. And the aim of the study was to determine whether the effects of NRPT treatment would increase regenerative capacity as measured by satellite cell function in humans. So we set up a clinical trial together with Aarhus University and Aarhus University Hospital. I have a good collaboration with Professor Niels Jessen at this institution. So we took aged humans. They were healthy. We recruited people above 55 years of age. We treated them with NRPT or placebo, and we induced muscle injury using this isokinetic dynamometer and stimulating muscles to contract eccentrically to induce the damage. This is the study protocol, and the study was really spearheaded by Jonas Brorson Jensen, who was a PhD student with Niels. So we had 32 participants, both men and women, and they went through either placebo or NRPT supplementation trial. 2 weeks prior to the muscle injury, we started treating them with NRPT, and then you can see here just an outline of the different events in the study. So we took multiple biopsies, both 2 weeks before, immediately before the muscle injury, 2 days after, 8 days after, and 30 days after. So multiple biopsies in both legs, actually, both the previously injured leg and also the control leg. We did a strength test, MR spectroscopy, and also measured the activity of the people. So this is just a video showing the intervention, the muscle damage. So you see you contract the muscle using these patches, and then you basically, so that will of course stretch your leg, and then the arm here will extend the muscle while it's contracting. So it's an eccentric muscle contraction, and that's very damaging to the tissue. And you can also see this on the MR spectroscopy. We can see a large edema in the vastus lateralis muscle here compared to the non-stimulated leg. So, so we could also see based on blood measurements, we have creatine kinase and myoglobin levels increased quite dramatically over the course of the study. So immediately after, or at least 2 days after, we had high levels of creatine kinase indicating muscle damage. And based on FACS sorting of cells that we did on the muscle biopsies, we could also see that we had many more immune cells or CD45 positive cells peaking at here day 8 of the study. So the model seemed to work. How about did they actually obtain take up the NR and pterostilbene? We can see increased levels of NAD in whole blood here at both immediately at the time of damage. So this is 2 weeks of treatment and also 8 days after. And in AAD and methyl levels of so NAD has been broken down to nicotinamide, and then nicotinamide is methylated, and there's a further breakdown, and the methyl 2-PY and 4-PY are breakdown products from nicotinamide. So that's indicating that they have had an increase in nicotinamide levels, at least in the cell or in the body. We also measured sulfonated pterostilbene, and we could see that being increased in the blood, and also the muscle. I don't show the data here, but this is sulfonated pterostilbene was also increased in the blood, at least. So these are the main results, and so we measured satellite cell number and also the size of the satellite cells, and you can see that there was an increase in satellite cell number over time, and so there was also an increase in size of the satellite cells with time, but there was no difference between the placebo and the NRPT-treated group. We isolated the satellite cells and we stimulated them or gave them EDU to measure the capacity to proliferate, and we did that over the course also of the study. And although there's a 0.051, I think, p-value for the interaction here, but it doesn't come out significant. But if anything, it actually seems like the NRPT group has a little bit lower capacity to proliferate compared to the placebo group. We also, on histological slides, we measured the centralized nuclei number and also the expression of the embryonic myosin heavy chain, and we can see that the centralized nuclei is actually only increased at the later time point, and so are so is the expression of the embryonic myosin heavy chain. So another thing that we can measure is the muscle fiber size distribution, and the mean fiber area. We can see that that declines a little bit, indicating that in the damaged, this is only in the damaged leg, of course, or the damaged biopsies. But we can see that we have a little bit more smaller fibers at the end of the study. We can also see here that the fiber size distribution actually is similar across, so there's no difference between the placebo and the NRPT treated groups. So no effect of the treatment. We can also see that it appears as though the NRPT group has a little bit smaller fibers to begin with. They were actually significantly older, although we tried to randomize for this. Age was a factor that came out to be significant between the groups, and that that may also impact the results, but it wasn't. I mean, I think the placebo group was 63 years of age, and the NRPT group was 69 years of age, so it's not a huge difference. But, yeah. Finally, muscle contractile function after injury is not affected. It's affected by the. You can see that compared to the value, it's reduced, but there's no difference between the contractile force. This is maximal voluntary contraction at 2 different angles, and we also measured rate of force development in the muscle, and there was also no difference here between the placebo and the NRPT treated groups, although the rate of force development in the muscle, and this is the whole skeletal muscle, not only the lateralis muscle, but it was significantly reduced. So, to sum up, I think we have a very nice model to study contraction-induced muscle injury. It seems to work the way it should, but dietary supplementation with NRPT does not seem to affect any of the parameters that we measured in this clinical trial. So with that, I'll end here and just acknowledge people who have been contributing to the work. From my group, Emilie, Sabina, and Mads has particularly contributed. And then, as I mentioned, Jonas has really been spearheading this project together with Jean Faub in the lab of Niels Jessen. So thank you very much.
Thank you so much, Jonas, for that excellent but depressing talk.
There is one other paper testing this NR-pterostilbene combination in humans, and they reported an increase in LDL cholesterol. Did you also look at LDL cholesterol in your group?
In this study, I don't believe we did actually. No, we didn't. And in the previous clinical trial we did. We only had NR. We didn't do NR and PT. So I can look up whether we. But it's a good point, and I know that there is some concerns with basis regarding the cholesterol. But
it's probably related just to the pterostilbene, not to the NR.
Yes, exactly.
Terrific. By the way, we could never do it in the United States, this study, I have to tell you. We're kind of jealous or upset, I don't know how to say it, but look, first of all, from an NAD perspective, you had few responders. Okay, I wonder if they did better, but also when you had the 2/4, the metabolite turnover, what do you learn from that? I mean, does it mean that you give the NAD but it just doesn't go to where you want it to go? I mean, what would be the biology that explains the increased turnover without any increase in the...
No, I think it's fair to say that much of the... So we gave 1 gram of NR per day, and much of that is degraded to nicotinamide. And the body will try to get rid of that because that's probably very excessive. So much of it will be turned over to nicotinamide, and then that be degraded. And the levels of 2-PY and 4-PY, it's basically indicating that the nicotinamide is being degraded and being ready to be excreted in the urine. So in the other clinical trial we did, we also measured these metabolites in the urine. We can see sky-high levels of methylated 2-PY and 4-PY in the urine. After that was a 2 gram of nicotinamide riboside, so even higher levels of the metabolite. But yeah.
Interesting. Can I ask a question, maybe? So maybe I missed it. What was the age of the subjects in this study?
Yes, I mentioned quickly, and I didn't put it on the slide, but I think the placebo group averaged 63, and the NRPT was 69 in average, I think.
Okay, so it was in elderly, actually.
Sorry?
It was in elderly individuals.
Yes. Yeah.
I have a question here.
Sometimes in studies you get negative signal because of dose. Would it be worthwhile, in your view, to do a dose ranging study where higher, likely non-toxic doses of this protocol were used?
I definitely think that there are some studies in rats and also in mice showing a dose-dependent effect on specific. I can't remember the endpoint, but there are at least studies indicating optimal doses that are much lower in animals at least. And it could be interesting to do those kind of studies in humans as well, and see whether, I mean, you could speculate that we're just giving too much, and then we, for instance, deplete methyl donors, or if you want to, if the body needs to methylate the excessive nicotinamide, for instance, then those methyl groups will not be available to for instance, manipulate or regulate chromatin, for instance, right? So there could be downsides of having too much, giving too much.
Very interesting. Pekka, last question.
Thanks, that was very nice and thorough. Of course, there's 3 stages on the repair by satellite cells. You have to activate them, they need to proliferate, actually then fusing into myofibers. So the fact that you had more proliferating but not any positive effects, did you look at the end stage, the fusion of the myoblasts into forming fibers?
Yeah, no, we didn't do that, but this is something that we're actually following up on. We're trying to dissect, if nicotinamide riboside supplemented to cells in culture, what stage would be beneficial for the process of differentiation and fusion or proliferation. So we're doing those studies right now.
Very cool. Thank you so much, Jonas.